Good morning. So as my title slide shows, mitochondria in this slide, in this picture, is labeled in red and orange. The point of this picture is to show you that if we label mitochondria, this is a mouse proximal tubule epithelial cell line. We see that the mitochondria actually is transient. It moves from one cell to the other through these tube-like structures, and also mitochondria is released. So from this picture, the idea that mitochondria could be used as a therapy is something that was not far-fetching. So the outline for the talk today is I'm going to talk about role of mitochondria in AKI, its role that pertains to its function beyond making ATP, methodologies that are currently available for detection of mitochondrial damage as it relates to AKI, mitochondrial DNA as a potential biomarker, and how it correlates to disease severity. And talk about the therapeutics that are currently available. Most of these are preclinical. However, there are some clinical trials going on now. Pharmacological, cellular, for cells, it's stem cells that act as mitochondrial donors, and then mitochondrial transplants, and I'll get into that a little bit later. And the idea of mitochondria transplant is termed mitoception. So there's no need for introduction, but I'll quickly go through this slide. AKI is the abrupt loss of kidney function, which results in uremia and the dysregulation of extracellular volume and electrolytes. AKI may rise from multiple initiating insults such as ischemia, IRI injury, cardiovascular surgery, contrast administration, and sepsis. AKI itself is an independent risk factor for CKD and end-stage renal disease, and that severe AKI can be accompanied by an irreversible decline in kidney function. There are no effective treatments available, and therefore the best treatment is prevention. This slide, even though it's a little busy, it shows you that mitochondria in a normal, healthy proximal tubule is found in this complex network. It's linear in shape. However, after a injury, either a toxin, inflammation, or ischemic-induced injury, this mitochondria now swells up. The shape of it changes to a bigger shape. And this swelling results in leaking of mitochondrial DNA into the cytoplasm and also into the extracellular space. And overall, there's decrease in ATP function for obvious reasons. And this leads to eventual cell death in the kidney. Currently, there are no drugs or compounds that can block this effect. Rapamycin, through its ability to induce mitophagy, has been shown to have an effect on mitochondria and slowing of AKI. But if you can find a way to induce the master regulator of mitochondria biogenesis, which is PGC1 alpha, it's thought that this could prevent and block AKI and also block the progression to CKD. So mitochondria not only produces cellular ATP in the form of, energy in the form of ATP, but also modulates several cellular functions, including proliferation and intercellular calcium homeostasis. Mitochondrial dysfunction induces apoptosis and the generation of reactive oxygen species, ROS, both of which contribute to development and progression of various diseases, including AKI. As you can see in this diagram, mitochondrial damage is at the heart of most kidney-related injuries, have it be ischemia, toxin injury, or inflammation. After heart, kidney is the organ that expresses a lot of mitochondria. So any sort of insult to the kidney, mitochondria is involved. So in that regard, any sort of intervention that can keep the mitochondria intact, functionally and structurally, is beneficial to prevent AKI and also prevent the progression to CKD. So in ischemic injury or even in sepsis, mitochondria swells up, as I already showed you earlier, and this swelling up causes eventual loss of ATP. That's the biggest culprit to why a cell dies. obviously there's a release of cytochrome C and mitochondrial DNA, but this loss of ATP and loss of energy results in changes in how the cell looks and behaves, and eventually it causes death in the form of necrosis, and it induces inflammation through the release of mitochondrial DNA that can activate toll-like 9 receptors, and this causes AKI. There are other kidney-related diseases that also affect the mitochondria, but the mechanism of how that works is slightly different. And even in progression to CKD with hypertension, diabetes, or obesity, mitochondria is involved, and it's due to loss of ATP. This is a very busy slide, but I wanted to just point out a few players that are critical for mitochondria. One of them, as I mentioned, is called PGC1 alpha. It's a master regulator of mitochondria biogenesis. In preclinical studies with transgenic mice, overexpressing this gene can prevent AKI completely. Either a global knock-in or knock-in only in the proximal tubules. There are certain genes that are involved in causing the mitochondria to become shorter, fission, or causing the mitochondria to become longer, fusion, which are DRP1 and OPA. And eventually, mitochondria is involved in mitophagy as well, which is important for proliferation. And in the mitochondria, you can see that the mitochondrial DNA is contained in the mitochondria, but upon injury, this mitochondria is released and induces inflammation, which, as I mentioned already, with the combination of other things, causes AKI. So clinically, is this relevant? In the urine, mitochondrial DNA has a strong correlation to severity of AKI. As the slide shows here, urinary mitochondria was measured by looking at this mitochondrial gene called COX-3. In patients with no AKI, depending on mild or severity, there's a positive correlation to the amount of mitochondrial DNA that's released. Why is that important? This correlation only is true for urine mitochondrial DNA. In the same patients, if you look at the blood mitochondrial DNA, it does not have a correlation to AKI. So in that regards, urine mitochondria DNA can be used as a biomarker to look at severity of injury and also if a therapeutic on board is working. So the current mitochondrial therapeutics, as I already mentioned, are pharmacological. There are a whole group of SS peptides, MitoQ, resveratrol, cellular in the form of mesenchymal stem cells as mitochondria donors. And I'll finish my talk by showing you organelle transplants, where you isolate mitochondria and inject, and then look for various means of how it's working. Once again, this is a busy slide, but it summarizes all the pharmacological data that's available in AKI. Majority of these drugs, like MitoQ, in this first paper, and also transgenic mice that are missing the DRP gene, or cells in mice that are given mesenchymal stromal cells that actually donate mitochondria, all of these therapeutics cause prevention of AKI. They have to be given prophylactically before the injury. All of these drugs that I showed you have been used in many of the preclinical models, from small mice to as big animals as pigs. And all of these result in preserving mitochondria function, which causes preservation of kidney function and eventual no injury and no AKI, therefore no progression to CKD. Clinically, these drugs are being tested in various models, but for some reason, AKI is not primary focus. Most of these drugs are being tested in models of CKD. Cellular interventions for mitochondria. There is limited data in AKI. There is many papers that show how mesenchymal stem cells are able to protect from lung injury. But the only data that's available in kidney is a current paper that just came out showing that the mesenchymal stromal cells actually preserve renal tubular cell mitochondria. But there was no evidence that these cells were actually donating their mitochondria. So there's another factor that is involved. For mesenchymal stem cells, there is currently 3 therapies for clinical trials, and these are all looking at AKI. So what are the limitations of these 2 therapies, pharmacological and cellular? That there's obviously off-target effects. There's off-site cellular engraftment with these stem cells, and the preparation that's involved in cells is very tedious. It takes a long time. You start with a limited number of cells, and it requires preparation ahead of time. And in certain cases, that can be immunogenic. So what about mitochondria transplants? Can that be done? In the heart, it's very easy, because in the heart, you're able to inject at the site of ischemia that was identified. So therefore, for the heart, they call it mitochondria transplants, because they're actually injecting it into the heart. So many preclinical studies. However, this has been done in humans as well, in pediatric patients who were born with a congenital heart defect. In those patients, the area was identified in the heart that was ischemic. Mitochondria was isolated from their pectoral muscle, injected at the site of ischemia, and by echo, a week later, that area was no longer ischemic. So in the kidney, there is no literature at the moment that shows that mitochondrial therapy could work. So as a reminder, you can see that after ischemic injury, there's a loss in tubular morphology. The cells die, the mitochondria is released, there's decrease in ATP, and eventually this all results in AKI. The electron microgram shown at the bottom shows you compare to a sham-operated mouse, even after 15 minutes of reperfusion, this mitochondria swells up. Excuse me, the quality is not as great as I hoped. So how can we look at that in a live animal? To test that, we actually made a mouse that had proximal tubule mitochondria that had a label associated with it. So we can look at immunofluorescence, and we didn't have to go at electron micrographs to look at the function. So we can see in this mouse, these 2 slides are from a sham-operated mouse, and this is just a bigger slide from the top slide. You can see the mitochondria is nice and linear. It looks happy. The tubular shape is intact. And after ischemia, once again, we're looking at this region blown up here, there's loss of mitochondria. And this signal that's associated with the mitochondria can be found in the urine, and it can be found in the blood. And eventually, it's picked up in the spleen. So it acts as a mitodamp. So that's great. Mitochondria isolation techniques take so long. How can this be clinically relevant? So we actually adapted a technique that was used in those patients, those pediatric patients I told you in the heart, which only takes about 30 minutes to isolate mitochondria. The isolated mitochondria is very pure. It's about 99% pure mitochondria, as shown here in the electron micrographs. And it makes a lot of ATP, shown here in blue. From these studies, we noticed that if you keep the mitochondria cold, they're very happy. They're structurally sound, and they continuously make ATP. But if you heat them at 37 degrees, they lose their structure, and they stop making ATP. So we actually use that to our advantage to test this in an in vivo mouse IRI model. So in these studies, we injected the 4-degree mito or the 37-degree mito a day before. Is an IV injection. Waited 1 day, did 26 minutes of ischemic injury. The next day, collected samples for function and histological analysis. And we can see that mice that received the mitochondria that was isolated and kept at 4 degrees, there was significant protection compared to the vehicle-treated mice. However, if you inactivate the mitochondria, this protection is lost. And histology follows the same trend as creatinine, where there's significant reduction in ATN in the mitochondria-treated mice that were kept at 4 degrees. Once again, not surprising, but mice that were treated with the 4-degree mitochondria have less cell death, as shown here in tunnel apoptosis. Their proximal tubules are preserved, which this is looking at the brush border with the CD13 antibody. And overall, this effect that's by the mitochondria is lost if the mitochondria is disrupted in function and structure. And also, mitochondria that's good results in less infiltration of inflammatory cells like macrophages and neutrophils. So we used the same mouse that I showed you earlier to see if our injected mitochondria in this mouse also preserves endogenous mitochondria. It's not a surprise, again, that if there's better kidney function, we should expect preservation of mitochondria, and that is the case. Compared to vehicle-treated mice, where you can see the mitochondria is swelled up, and most of this is released in the interstitial space, mice that receive the 4-degree mitochondria, there's preservation of tubular structure and endogenous mitochondria. So the million-dollar question is, you're injecting this IV, it's a systemic injection, does the mitochondria actually end up in the kidney? The mitochondria ends up everywhere, but yes, it actually ends up in the kidney in the proximal tubules. So we labeled the mitochondria with a purple dye, injected it, and then pretty much asked, where is it going? So we looked at all different kinds of organs. In the kidney, it loves to go into the proximal tubules. And the proximal tubules, once again, were identified with this brush border antibody, CD13. So I showed you that it works if you give it ahead of time. But what happens if you give it in an established injury model? So we used the same IRI model, but except we did a unilateral ischemia, and then the next day injected mitochondria, and then let the mice go for about 13 days, did a contralateral nephrectomy, control of the kidney, and then day 14 did the analysis. Even if you give mitochondria 1 day after injury, it seems to slow the progression of disease to CKD. There's reduced expression of fibrotic markers like SMA and fibronectin, and histologically, the kidneys look better. They're not great. Functionally, there's significant protection. Histologically, we see that there is less collagen deposition. So what are the requirements for this mitochondria? Well, I showed you, it has to be intact and it has to be viable. If you heat and activate it, it doesn't work. So it has to be respiration competent. Mitochondrial products like the DNA, RNA, protein, and complexes, or even ATP by itself does not do the same thing. So it has to be mitochondria. It has to be freshly isolated, but that's okay because it takes about 30 minutes to isolate it. Therefore, it's feasible for clinical use. So what is it doing? So in our lab, we noticed that if you label mitochondria before injury or after injury, it's different where it is found. So in a control setting, it's pretty much spread out everywhere. After you treat with LPS or do hypoxia for about 3 hours, it seems to move closer to the nucleus. So it seems to have a role where it interacts with the nucleus to induce some things to happen. So there's some literature now that shows that mitochondria actually moves so it can participate in things that are required for transcription, either good genes or bad genes or inflammatory or protective genes. At the moment, we don't know what those genes are. So in a cell culture, I showed you that it gets into the cell. Can we measure what it's doing in the cell? So in the cell, by using a machine called Seahorse Analyzer, which allows us to look at respiration of the mitochondria, we can see that compared to the white bars here, which were just normal cells, there's a dose-dependent increase in oxygen consumption. So that means whatever we're adding is consuming oxygen and making more ATP, which from this graph, we're able to calculate all of these things out. So we can see there's an increase in basal oxygen consumption, and there's an increase in ATP. So it's working. It gets in, and it's making what it's supposed to do, ATP. Well, from this image, you can see that the amount of mitochondria that get into the cell, it's very selective. Not every cell has the same number of mitochondria that get in. So how do we have such great effects? How do we protect from ischemic injury when only a limited amount of cells take up the mitochondria? The answer is PGC1 alpha. So what happens is when the mitochondria get into a cell, they activate this gene. And as a reminder, PGC1 alpha is the master regulator of mitochondrial biogenesis. So in a way, taking up healthy mitochondria kickstarts the cell to make more mitochondria of its own, and which is shown here. So there's a dose-dependent increase in PGC1 alpha transcript protein, and over time, this is after 24 hours of incubation, we can see the mitochondria DNA content actually increases, and these cells proliferate faster after you add mitochondria on them. So this could explain why we have not as much, in our post-treatment models, why it doesn't progress to CKD to the same degree. So if PGC1 alpha is important, what happens in its absence? Nothing happens. So it requires endogenous PGC1 alpha to have a positive effect, because if we knock out the PGC1 alpha, shown here in red, you can see it's very similar to the cells that got no mitochondria. So alone, it's not able to do much. It requires endogenous PGC1 alpha for inducing protection. So to wrap up, what are the advantages of using mitochondria for therapy? Well, it's fast. So if you know somebody's going in for surgeries, you can take mitochondria, which is autologous in nature, from a muscle biopsy, which is only about a 6 millimeter biopsy is required for mitochondria. So it can be done in the same location. It doesn't have to be a company that transports it over. It can be from self, so there are no immune responses. And as I mentioned already, it requires very, very small amounts to get large mitochondrial numbers, and it regulates itself. So once you add it, it kickstarts its own mitochondrial biogenesis. In addition to prevention, isolated mitochondria can be used in established injury to suppress the progression to fibrosis. So to conclude, uptake of healthy mitochondria by tubular cells and other cells helps maintain bioenergetics, maybe in the form of ATP, to help prevent injury and also to accelerate recovery. And I'd like to just finish by acknowledging my lab members and my research funding. Thank you for your time.